Welcome to Biochamp Academy. I hope you all are doing great. In this video, we are going to learn about the structure of the DNA. I have covered all the details about the structure of the DNA so you can clearly understand the chemical structure of the DNA. So let's begin with the video.
So let's begin with the structure of the DNA. Now we all know that DNA is a genetic material. It carries genetic information. So basically, let's take a simple example that we all human beings are having different eye colors, right? Some people they are having black eye color, some are having blue, some are having brown and some are having green. So who is going to determine the eye color? So basically this genes present inside the DNA is going to determine or going to decide the eye color of an individual. That's the reason the DNA is called as a genetic material that carries the genetic information of an individual. Clear? Okay. Now, whenever in exam they ask you about the structure of the DNA, like draw the structure of the DNA, right? So we know the simple structure of the DNA. Whenever we hear the word DNA, we know that DNA looks somehow like this, correct? But actually, they are not asking us to draw this particular structure. What they want us to draw is the chemical structure of the DNA that we are going to learn after a few minutes, right? But this structure is also equally important because it helps us to determine or to decide that DNA is double stranded as well as it is helical, right? So from this structure we can say that this DNA is double stranded and it is having the helix, double helix basically. Clear? Right. Okay. Now let's move towards the polymer and monomer concept for the DNA. So we all know that DNA is a polymer. Now you will say what is polymer. So if I think you must have the idea of the polymer, but if you are not having any idea, then let me explain you that polymer, poly stands for many, mer stands for units. So when many units of monomers, mono stands for single and mer stands for unit. So when single units are joined together, then they will make the polymer of particular molecule. So DNA itself is a polymer which is made up of monomers of nucleotides. So 1000 to 2000 number of these nucleotides will join together to make one particular molecule, one particular polymer of DNA. So this DNA is a nucleic acid, right? Nucleic acids are made up of nucleotides. So here I have simply explained you that these are the single units of the nucleotides that are the monomers. They are going to join up and they make the polymer of your DNA, right? So this is the basic information, or you can say basic explanation for the structure of your DNA, right? Now we have to know that what is present in this particular single nucleotide, that is called as the chemical structure of the nucleotides. So before directly learning the structure of the polymer of DNA, we first learn the structure of the single monomer or single unit of the nucleotides, because these single units are going to join up to make the final DNA molecule, right? So let's learn about the structure of the nucleotide. So here is the structure of the nucleotide. Now, nucleic acid is made up of nucleotides. So RNA and DNA both are the example of nucleic acid, right? Both are made up of nucleotides. So here is the reference structure for the nucleotide for RNA as well as DNA. So when they will give you the full structure of the RNA or the full structure of the DNA, then how to distinguish that whether it is a DNA nucleotide monomer or RNA nucleotide monomer, that also we are going to discuss in this particular structure, right? So first of all, we have to know that nucleotide is made up of three main elements. The first element is this one, which is the ribose sugar or deoxyribose sugar. Commonly they are known as pentose sugars. So this is the pentose sugar because it is having 5 carbon, carbon 1, carbon 2, carbon 3, carbon 4, and fifth one is here. That's why it is called as pentose sugar. Another element is the phosphate group, which is PO4 3 minus. So here I have written also it's PO4 3 minus. Now remember students, as I told you before, that DNA is made up of 1000 to 2000 number of these nucleotides, meaning it is going to have this 1000 to 2000 number of this phosphate group, right? And ultimately this phosphate group is PO4 3 minus. So it's the negatively charged group. Because DNA is having so many phosphate groups, ultimately DNA is also becoming negatively charged. Clear? So DNA itself is a negatively charged molecule. Okay, so this is about the first element, pentose sugar. This is about the second element, phosphate group. Now the third element is the nitrogen containing base. So this is the nitrogen containing base, right? Now this nitrogen containing base can be any out of this all. But if we talk about the DNA, then DNA can have only these four nitrogen containing bases: A, T, C, G, adenine, thymine, cytosine, and guanine. But if we talk about RNA, then in case of RNA, this T is going to be replaced by U, right? So RNA is not going to have thymine, it is only have uracil. Clear? This is the first difference between the DNA and RNA in terms of nitrogenous bases. Definitely RNA will have other three bases, A, C, G, but it is not going to have T. Whenever there is a T, it is going to be replaced by U, which is uracil for RNA molecule, right? So T is replaced by U for RNA. Another difference is DNA is double-stranded, RNA is a single-stranded molecule. So these are the two differences between the DNA and RNA. First one is T is replaced by U. Second one, RNA is a single-stranded molecule. Clear? Okay. So these are about the three elements which are present in a particular nucleotide: phosphate group, pentose sugar, nitrogen bases. Now, from the this pentose sugar, from the carbon number 1 here, it is always attached with this nitrogen base, right, by forming a covalent bond. So make sure, student, you have to remember this as well, that by which bond in a nucleotides this pentose sugar, phosphate groups, and nitrogenous bases are connected. So it's a covalent bond, right? Here also, so carbon 1, 2, 3, 4, and 5th. So from the 5th carbon, it is attached with the phosphate group. So this is also the covalent bond. So this both are going to be the covalent bonds. Okay, in some schools they used to refer the term covalent bond as a phosphodiesterase bond. So it's up to you. You can remember it as a covalent bond also, and you can remember it as a phosphodiesterase bond also. Clear? Okay, so within a nucleotide, all of these three elements are joined together by forming covalent bonds. Okay, now we have covered about the phosphate group that makes our DNA molecule negatively charged. We have covered the nitrogenous bases, that we are having four different kind of the nitrogenous bases: A, T, C, G. Now another thing we have to know about this nitrogenous base is here, that out of these four nitrogenous bases, two are having purine ring in their structure and two are having pyrimidine ring in their structure. So what is the difference between this purine ring and pyrimidine ring? So purine ring are always having this kind of structure, 6-member ring attached with 5-member ring, while pyrimidine ring is only 6-member ring. So it is having carbon 1, 2, 3, 4, 5, 6. That's why 6-member ring. Here 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5. So 6-member ring is attached with 5-member ring. It is purine ring. Only 6-member ring, it is pyrimidine ring. Apart from carbon, they do have nitrogen because it's a nitrogen containing base here, 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 here. But it's okay, student. We are not discussing here chemistry. We are learning about biology. So no need to remember the position of the nitrogen. But you should be very clear that Ag, adenine, and guanine, these two are having purine ring in their structure. TC, thymine, and cytosine, these two are having pyrimidine ring in their structure, because many times in MCQ they are asking this question. I hope that now you are clear with the purine and pyrimidine ring differences and which bases are having purine ring structure and which are having pyrimidine ring structure as the nitrogenous base. Clear? Okay. Because they are having this purine ring and pyrimidine ring in their structure, there is a complementary base pairing rule between these bases, that because of the structural difference, A is always paired with T and C is always paired with G. Adenine to thymine, cytosine to guanine, right? But if it is RNA, T will be replaced by U. So this is called as complementary base pairing rule. Who developed or who found this complementary base pairing rule? Who found the structure of the DNA? That all we are going to learn in part 2 of this particular video series for the DNA. So make sure, student, you keep an eye on the Biochamp Academy's YouTube channel. Soon I am going to upload a new video about the theories, that who found the structure of the DNA, what is nucleosome, how DNA can get replicated, what is transcription and translation. Okay, so coming back to the today's video, the pentose sugar. Now we have to learn about the pentose sugar. Now, pentose sugar can be of two types. It can be the ribose sugar or it can be deoxyribose sugar. So if I ask you the full name of a full form of DNA, you will say deoxyribonucleic acid. Why? Because DNA is always having deoxyribose. That's why deoxyribonucleic acid. While RNA is always having ribose sugar. That's why it is ribonucleic acid. Clear? So the third difference between DNA and RNA is RNA is having ribose sugar as a pentose sugar. Clear? So here I have drawn the structure also for the ribose and deoxyribose. So if I'm not going in detail about these two structures, because I have already covered this structure in part 3 video for molecular biology, so molecules to metabolism unit. So please watch that video as well. I'll put here the link as well as in the description, where I have given some tricks and tips so you can easily remember the structure and differentiate the structures between ribose and deoxyribose. Okay, so this is about the structure of the nucleotide. Again, let's do a quick revision. Nucleotide is made up of pentose sugar, phosphate group, nitrogenous base. They all are joined together by forming covalent or phosphodiesterase bonds. Phosphate group makes the nucleotide or the entire DNA structure negatively charged. Pentose sugar can be of any two type, ribose or deoxyribose, and nitrogenous base can be any out of these four for DNA, ATCG. They can have purine ring in their structure or pyrimidine ring in their structure. Adenine and guanine, they both are having purine ring, while thymine and cytosine, they both are having pyrimidine ring. There is a complementary base pairing rule in which A is always complementary base paired with T and C is always pairing with G. Clear? So this is about structure of nucleotide. Now let's see about the structure of DNA. So DNA is always double helical, right? It is double stranded and it is antiparallel. So one strand of the DNA will be antiparallel to the other strand of the DNA. So let's draw now the nucleotide. So when they ask you for 7 to 8 marks about the structure of the DNA, then you have to draw 4 nucleotides on one strand and 4 for the other strand. But when they ask you it for 4 marks, then you have to draw 2 nucleotides for one strand and 2 for another strand. Okay, so here I am going to draw the 4 nucleotides and also I am going to label it, and I'll show you how you have to label to get the full marks, right? So let's start the drawing for the structure of the DNA. So first is the phosphate group. This phosphate group is attached with the pentose sugar. So we are drawing here the pentose sugar, right, on the 5th carbon. Then it is going to attach with the nitrogenous base. So this is going to be the first nucleotide. Another nucleotide, same, we are going to have this phosphate group, then this pentose sugar, and then this nitrogenous base. So make sure, student, you have to draw it again and again. You have to practice it, then only you can draw it, very easily, very quickly. So you have to draw it for 4 times because we want to show 4 nucleotides like this. So this is my 4th nucleotide, right? Okay, so this is the one strand of the DNA. Clear? Okay, so I am now going to draw the another strand for the DNA. Now another strand is going to be antiparallel. So nitrogenous base here, because we know that nitrogenous bases are having complementary base pairing and they are attached with each other. That's why the another ring is antiparallel. So this nitrogen base can come in contact with this nitrogen base and can form a bond. Imagine that instead of this nitrogen base, here is a phosphate group and this strand is also parallel. This phosphate group cannot make the bond with the nitrogen base of the first strand of the DNA. Make sense? That's the reason that another strand of the DNA is antiparallel. Okay, so now let me draw the nucleotides but antiparallel for the second strand like this. Clear? Right? Okay, so this is my first nucleotide. You have to remember the numbering, student, that nitrogenous base is always attached with the first carbon of the pentose sugar like this, and the phosphate group is always attached with the fifth carbon, right? Like this. This is the third one. We have to draw total 4. I am drawing it so that you can also refer that how to draw. That's why I am drawing it live. I have not used here the already drawn structure, right? Okay, so this is about my last nucleotide. So these are the 4 nucleotides for this particular strand and these are for another. Now we know that these are joined together by which bond? That we have already discussed in the structure of the nucleotide, right? So you will say covalent bond. So it's correct, they are joined by covalent bond. Now they are still not attached with each other, right? These all are separate, they are not attached, they have not make yet the strand of the DNA. How they are going to attach? So remember, student, from the third carbon, first carbon, second carbon, third carbon, here is the third carbon, they are going to attach with the phosphate group. From the third carbon, they will be attached with the phosphate group. From third carbon, attached with the phosphate group. Clear? So this is how they are going to attach. Clear? Phosphate group and third carbon. Again, this bond is also a covalent bond. So make sure, student, that within a nucleotide or within a single strand of DNA, all elements as well as nucleotides are joined by covalent bond. Important MCQ or 2-mark question. Clear? Okay. Same way for the second strand. Here also, this is going to be the third carbon. This is the third carbon attached with the phosphate group. Third carbon, because this is first, second, third, right? And third carbon attached with the phosphate group. Clear? So these are also the covalent bond. Another thing we have to remember here is, student, see here the fifth carbon, this is the fifth carbon which is attached with the phosphate group, right? The fifth carbon attached with phosphate group. That's the reason this is 5' of this strand. Here is the third carbon which is facing downside. That's the reason it is going to be the third prime. So this is going to be 5' to third prime. Let's see for the opposite strand. This one is the third carbon facing to the top. That's the reason this is going to be third prime. Fifth carbon, this one is facing down, so 5'. So see, 5' to third prime, antiparallel, third prime to 5' antiparallel. Clear? Yet these two strands are not joined with each other, right? There is no bonding here. So in DNA, they are also joined together. How? So let's see here. First, let me write here the name of the bases. For example, here I'll write A, C, G, T. What would be complementary? So, A to T, C to G, G to C, T to A. Another important tip here, student, make sure you always write full names of your bases. Otherwise, they won't give you the mark for A, T, C, G, okay, for the short forms. So you have to at least for one time in your answer write the full name and then you can use the abbreviated version, okay? Okay. Now, how this A will join with the T? So this A is always joined with the T by not forming covalent bond but forming hydrogen bond. So the two strands of the DNA are joined together by forming hydrogen bond. Within the strand of DNA, nucleotides are joined together by forming covalent bonds. Another thing, see here I am drawing 3 dotted line for C to G. Again, C to G, 3 dotted line, right? And for T to A, 2 dotted line. Why? Because according to the structure, 2 hydrogen bonds are formed between A to T and 3 hydrogen bonds are formed between C to G. So here they are joined together by hydrogen bonds. Hydrogen bonds. Right? When they ask you for 4 mark, you have to draw, or 8 mark, you have to draw this entire structure with the labeling. So apart from this labeling, you have to label it that this is the nitrogenous base thymine, full name. You have to label it, this is the pentose sugar in the form of deoxyribose, this is the phosphate group, right? We have already labeled it in the structure of the nucleotides. You already know that, so I'm not labeling again here, but make sure you label it, right? So this is the clear structure of the DNA, and with the clear explanation that which bonds are there within the strand, between the strands, how the complementary base pairing are formed, the rule of the complementary base pairing basically given by the Watson and Crick scientists, that we are going to discuss in the next video. Clear? So we are keeping up to this for this particular video. I hope that now you are clear with the structure of the DNA. And thank you so much for watching this video and learning with Biochamp Academy.
Please like the video, share the video with your friends and subscribe to the channel. I will see you soon with another interesting topic of the biology. Thank you so much for watching this video.